In the previous video, we looked at the first hybridization experiment that Gregor Mendel did involving a purebred smooth-seeded pea plant hybridized with a purebred wrinkled-seeded pea plant, and we saw, as Gregor Mendel did, that only smooth-seeded pea plants were produced because the smooth-seeded allele is dominant to the wrinkled-seeded allele. Now, because each gamete that produced these offspring contains only 50% of the DNA, that means that the smooth-seeded parent contributed one allele that codes for smooth seeds, and the wrinkled-seeded parent contributed the other allele that codes for wrinkled seeds, but we don't see the wrinkle-seeded allele because the smooth-seeded allele is dominant over the wrinkled-seeded characteristic. And what Gregor Mendel did next is he took two of the offspring plants that were created as a result of this first hybridization and did another experiment, which is what we call a monohybrid cross. So we can refer to each of these as being hybrid plants because they contain one smooth allele and one wrinkled allele, one dominant and one recessive allele, meaning that neither of these plants, even if they have the dominant characteristic, neither of them are so-called purebred, and we'll come up with a term for this. We call it a monohybrid cross because Mendel was looking specifically at the smoothness or wrinkledness of the seeds, meaning that we are only looking at one characteristic. Now, what we can do is, if he took two of these offspring plants here, we can use a Punnett square in order to predict what kind of offspring these offspring in turn are going to have. So because of the law of independent assortment, Gregor Mendel was actually the first biologist to coin this term, even if he didn't know the biological basis in meiosis, why this happens. The law of independent assortment dictates that when a plant or any sexually reproductive organism makes gametes, they can only give one allele of the two alleles that they have, meaning that one parent in the pairing has the dominant allele or the recessive allele, but because gamete production involves only 50% of the parent DNA, this parent would only be able to give the dominant allele or the recessive allele and not both. And because the other parent had the same genotype, the same combination of one dominant and one recessive allele, the same would be true for them as well. And we could, using the Punnett square, predict what the possible genotypes of organism or offspring organisms would be, depending on which allele was carried by which gamete. So if this dominant allele was carried by the sperm and fertilized an egg also containing a dominant allele, we would have this zygote produced that would contain two copies of the dominant allele. Now, every one of the other possibilities we will see is actually distinct from this, because here we can see that this parent, in the case of this gamete, would contribute the recessive allele, but would fertilize an egg that contains the dominant allele, and so we would end up with the same genotype as each of the parents. Likewise, this fertilization event here, this parent up top, would contribute the dominant allele, and this parent here has an egg that contains the recessive allele, and so again, we end up with the same genotype as the parents. Now, all of these, because all of these organisms here all contain the dominant allele, which is smooth-skinned, all of these would show the smooth seed phenotype here, because as long as the dominant allele is present, it overrides the recessive allele, even if it is there. But with this fertilization event, if both parents contribute a sperm and an egg that contains the recessive allele, now we actually would see an offspring organism that actually has the wrinkled phenotype visible. And what we see from this classic monohybrid cross is that there is a very specific and predictable genotype ratio that occurs within all of the offspring. We would expect that 25%, meaning 1 out of 4 possible offspring, would contain 2 copies of the dominant allele, and likewise 25%, or 1 out of 4, would contain 2 copies of the recessive allele, and the remaining 50% would contain both alleles, just like the parents would. And this is exactly what Mendel saw when he actually got two hybrid pea plants to reproduce. These are exactly, or very close to it, the ratios that he saw in offspring with specific characteristics. And we actually have names that we can use for each of these different genotypes. For example, a smooth-skinned pea plant that has 2 copies of the dominant allele, or the dominant form of the characteristic of smooth seeds, is what we call homozygous. Homo, of course, is the prefix that means same, meaning that the zygote produced from this fertilization has the same alleles, which happen to be the dominant allele. We can also use the word homozygous to describe a situation where the pea plants only contain wrinkled seeds, only this time, instead of containing 2 copies of the dominant allele, now wrinkled pea plants, or wrinkled seeded pea plants, would have 2 copies of the recessive allele, and therefore be called homozygous recessive, while the situation involving both parents and a situation where the zygote contains 1 of each allele, because the dominant and recessive alleles are not the same, we refer to these as heterozygous organisms, because they contain both the dominant and recessive allele. There's just one more problem that Mendel had. Because the smooth allele is dominant to the wrinkled allele, as long as a smooth-seeded pea plant contained 1 copy of the dominant allele, we would see this phenotype here, regardless of whether the organism was homozygous dominant or heterozygous. So what we can see is that there are multiple possible genotypes that give us the same phenotype here: homozygous dominant or heterozygous. Now, obviously, we cannot see DNA, and Mendel didn't even know that DNA was the molecule of heredity. So how was he able to identify which organisms were homozygous dominant and which were heterozygous? This brings us to the last of the first 3 experiments that Gregor Mendel did, which is what we call a test cross. Let me just rewrite the word test more neatly before we explain what a test cross is. So once again, the problem is that Gregor Mendel was not able to distinguish whether a smooth-seeded pea plant was homozygous dominant or heterozygous. So what he would do in order to test this is he would take a smooth-seeded pea plant, regardless of whether he didn't know the genotype, and make it reproduce with a homozygous recessive organism. In other words, a pea plant that had only wrinkled seeds, and we will see that this produces 2 very different results. For example, if the smooth-seeded pea plant is homozygous dominant, it has no choice but to give a dominant allele, the smooth-seeded allele, and if we reproduce it with a homozygous recessive wrinkle-seeded plant, again, this has no choice but to give the recessive allele, and therefore, as we would expect, all organisms here would be heterozygous. So we can see in this case, because this is exactly what Mendel saw in the very first experiment, 100% of the offspring from this pairing would have smooth seeds, because every single one of them contains the dominant allele, and therefore we would see 0 offspring with the recessive allele. But when we try this with the heterozygous parent, because a heterozygous parent has one copy of each allele, it can either produce gametes with the dominant allele or the recessive allele, even if the other parent can only give the recessive wrinkled skin allele. However, if we use the Punnett square to check, these two offspring would receive the dominant allele here from one parent and the recessive allele from the other parent, and so each of these would be smooth-seeded peas, but because the parent is heterozygous in this case, these possibilities account for receiving the recessive allele, and so in these cases, both of these organisms here have no dominant allele, and therefore would have wrinkled seeds, meaning that in this situation where we cross a heterozygous organism with a homozygous recessive organism, 50% of the offspring would be expected to have the dominant phenotype, while the other 50% would have the recessive phenotype, therefore showing that we would get different offspring based on the phenotype depending on what the genotype is. And this is how Gregor Mendel was able to determine the difference between homozygous dominant and heterozygous organisms, even if he didn't know what DNA was. In the next video, we're going to look at what Gregor Mendel did to expand on his results, where he would actually look at multiple characteristics that his pea plants demonstrated and look at the inheritance of multiple characteristics at the same time.